Hello class, and welcome to Clinical Physiology. This is Dr. Lisa Carney Anderson, and I'm your instructor for this course. And this is our very first presentation of the class, and it's an introduction to chemistry and biochemistry. I really like to work with learning objectives. So here are some things that you ought to be able to answer after viewing this presentation and studying the material. Actually, what I would advise is taking a blank piece of paper, look at these learning objectives, and see how much you can write. So for example, be able to compare and contrast kinetic and potential energy. What are the definitions? What are examples of these two types of energies? Explain how the laws of thermodynamics impact how we think about biological systems. So energy is not created or destroyed, it's converted into different forms. What does that mean in terms of a biological system? We're going to be talking about a lot of molecules, and we have to have an understanding of the way molecules bind with each other. So hydrogen bonding, ionic bonding, and covalent bonding. Give examples, and then you'll have to apply. For example, when you put a molecule in water, how does the kind of bond influence how something interacts with water? We're going to talk about free radicals, and it used to be that we thought all free radicals are harmful because they zing around and damage molecules, but there are some free radicals like nitric oxide that are beneficial, so we want to talk about that. We want to talk about the concept of polarity. So molecules have a nucleus and they have an electron shell, and different atoms have differing amounts of electronegativity because of the electron shell. And so when there's an uneven distribution of that, how does that cause polarity, and then what's the consequence of polarity? We want to talk about what pH is, how pH allows hydrogen ions to associate with molecules, something we call protonation, and then how do levels of protonation affect how a biological system would work. But wait, there's more. What's an amino acid? What's its basic structure? How do they bind in a primary structure? Or how do they bond in a primary structure? How does the thing fold and form a secondary, tertiary, and quaternary structure? When we have a fully folded protein, we could have something that's an enzyme. What's an enzyme? How does it work? What does it do? Why is it essential to cell metabolism? Carbohydrates, what are they? Sometimes we need them for nutrients, sometimes we need them for structure. What's a lipid? And what are the three kinds of lipids? Triglycerides, phospholipids, and steroids. What are those doing in the cell? And finally, what are high energy compounds? And what do they do in the cell? And the major high energy compound we'll be talking about in this course, of course, is ATP. All right, energy. Energy is defined as the ability to do work. And we have two types of energy. So potential energy. Potential energy is stored energy. So you work really hard to pump the water up to the top of the hill. Water being at the top of the hill has energy in it. That's potential energy. Then we let the water flow down the hill, and as the water flows down the hill, it turns a turbine, which creates electricity. The water flowing through the turbine then is kinetic energy. That's energy in motion. We're going to see examples of this in the cell all the time. You store energy in a bond. You release the energy in the bond, and then you drive the transport of something. So we have couplings of potential and kinetic energy all the time. And when we think about these conversions of energy, we have to pay notion to the laws of thermodynamics. Energy is not created or destroyed. It's converted into different things. So we have energy in a donut, and then we eat the donut, and we break the donut down into its sugars, and we absorb that, and then we use the sugar to make ATP. So we're breaking the bonds in the sugar and harvesting the energy to then make ATP. Then we take the ATP and we break the bonds in the ATP and then we phosphorylate it and make a protein. Every time we're breaking bonds, every time we're forming bonds, we're converting energy. And when we convert energy, some of the energy is used to do work. We're pumping an ion. We're contracting a muscle. However, not all of that energy goes to the work we need to do in the cell. Some of the energy is given off as heat. So there's no such thing as a perfect energy conversion. We have to be aware of that when we think about biological systems. So here's an example of a atom. So this happens to be a carbon atom. So you see there are neutrons and protons in the center, and then we have 2 electrons in the first shell. And I'll get my pointer out here. So there's 2 electrons, green electrons in the first shell, and then there's 1, 2, 3, 4 electrons in the outer shell. So the electrons in green are associated with the carbon atom. Then we have 4 other atoms of hydrogen. Hydrogen has a proton and 1 electron. So those are the orange electrons. Whoops, I got to get back to my picture. So carbon is such a wonderful atom for biological systems because it has 4 electrons in its outer shell and is able to share those electrons with 4 other, what we call moieties, atoms or side chains. And then when these 4 atoms are then are sharing paired electrons with carbon, we say that carbon is then saturated. So here's our carbon, and then there's a hydrogen and a hydrogen and a hydrogen and a hydrogen. Carbon could share electrons with another carbon. And so carbon can form very, very long chains, and it's the basis of these long chains that allow us to have such complex molecules such as carbohydrates, proteins, and fats. Notice these are shared pairs of electrons, and therefore those are covalent bonds. Covalent bonds are the strongest of the bonds. Typically we need enzymes to form and break those bonds, so enzymes are controlling the metabolism of cell through the formation of these bonds. We have other bonds, ionic bonds and hydrogen bonds. So here are some ions, and here we have a sodium and we have a chloride. Dry sodium and chloride form ionic bonds. Notice if these ionic bonds are then put into water, then water can actually separate the sodium and chloride into those ions. So ionic bonds can be broken simply by putting in water. We don't need an enzyme. And then look what else happens. We have water, which is a very polar molecule. So there's the protons and there's the oxygen. Notice oxygen has a lot more electrons in its outer shell. It has more electronegativity. And then here we have the protons, and there's only 1 electron in the outer shell being shared here, and therefore water is very polar. One side of the molecule has more positivity, the other side of the molecule has more negativity, and therefore water is a wonderful substance to pry apart ionic bonds and then form layers of hydration around the ion. And then here you can see that one water molecule can have an association to another water molecule. So we see 2 kinds of bonds in this picture. We see ionic bonds between a sodium and chloride, and we're seeing hydrogen bonding between water molecules and between water and ions. Now, free radicals are those substances that have an unpaired electron in their shell. They could be negatively charged, but they don't have to be. And here we have 2 examples to show you what I mean here. So here's an oxygen molecule and an oxygen molecule. And 2 pairs of electrons are being shared in a double bond, a double covalent bond. This is an oxygen molecule. It's a very nonpolar molecule. Now over here you see we have, instead of having these 2 electrons here, we only have 1. And this forms a free radical we call superoxide. And superoxide is a good one to become familiar with in biological systems, because superoxide can be formed wherever there's high levels of oxygen. So those of you in the class who are clinicians have maybe been told that oxygen is a drug, and you give patients the amount of oxygen they need. You don't necessarily give them more. If somebody is okay on 60% oxygen, there's no reason to give them 100% oxygen. It also depends on your clinical site, and I'm digressing a little bit. But the point I'd like to make is that when you have lots of oxygen around, you can get reactions that lead to superoxide, and in the mitochondria, where oxygen is serving as the final electron acceptor, free radicals can be produced in the mitochondria. We're going to learn that the mitochondria have defense mechanisms, but whenever we think about oxygen, we can get what are called reactive oxygen species, and these are free radicals that come from oxygen. And because they have this unpaired electron, they want to react with something. They want their outer shell to be filled like this one. They want pairs. And so they will react with things. They'll react with proteins, and react with DNA, and react with fats. So if they react with protein, you can have a protein that doesn't work anymore. If they react with fats, you can have a membrane that starts to fall apart. If they react with DNA, you can have mutations that didn't used to be there. So free radicals can cause a lot of damage. Alrighty. Here are some molecules. Here we see the sort of the stick figure, and then we see more of the geometric formations. So methane is a very nonpolar molecule because the hydrogens are distributed. We have a distribution of electronegativity. Ammonia and water are a little more polar. Ammonia is a little more polar than methane, though ammonia is well able to get through membranes, so it's still, it's sort of in between water and methane. Water is the most polar, and again, as I've said before, we have a lot of electronegativity associated with this oxygen, less with the water, and so there's an uneven distribution of electric negativity across the water. So water, rather than ammonia or methane, makes an excellent solvent because of its polarity. Methane and ammonia are molecules that would be able to go through a lipid membrane because they're not, they're much less polar, they're nonpolar. So it's a good idea to be familiar with some of the side chains we can encounter in biological molecules and get an idea of the polarity or not. So carbon binding to carbons and chains, or carbons, I think I just said that wrong, carbons binding to carbon and chains, and then carbon chains with hydrogen groups, these tend to be nonpolar. So let's say we've got a fatty acid. I'll show you an example of that later on in the presentation. You just have carbon and hydrogen, long chains of that. Even distributions of electronegativity, and therefore these are non-polar compounds when you have a lot of this represented in your molecule. Now, when you have atoms such as oxygen or sulfur or nitrogen that have a lot of electrons in their shell, and then they bind to hydrogen or these R represents some kind of group. R stands for remainder group, but it just says there's more of the molecule than the picture shows. There's an uneven distribution. You see with the hydrogen and the oxygen, the sulfur and the hydrogen, the nitrogen and the hydrogen. So these are polar bonds. We wouldn't expect to see a lot of this in a lipid, but we would certainly expect to see these in proteins and nonpolar proteins. We have some side chains. These two side chains are very, very important to amino acids. So we have the carboxyl group. Notice this carboxyl group is ionized, so we have a negative charge here, but we could have a hydrogen. When the hydrogen is there, we'd say the molecule is protonated. Here we also have ionization because of protonation, so instead of an amino group, which is NH2, we have NH3+ because we have an extra hydrogen that's associated here. Phosphate groups introduce polarity. So if you have a phospholipid, you have a part of the molecule that's very hydrophobic, and you add a phosphate, and then you have a hydrophilic. You add hydrophobic and hydrophilic together in the same molecule, and you have something that's called amphipathic. So these are some side groups and whatnot to be familiar with as we're going forward. Again, here's the demonstration of the polarity of oxygen, the uneven distribution of electronegativity, and the ability of water to form hydrogen bonds. This can give some structure to water, and we see this in the phenomenon of surface tension. So for example, you fill a glass up to the very tippy top. There's a dome or a meniscus on the top of the glass. That is the product of water molecules forming hydrogen bonds and having some structure that supports that dome structure at the top of the glass. So when we have covalent bonds, we usually need an enzyme and an enzyme reaction, and we have a series of reactions in organic molecules that require the removal of water as we form the bond. So here we have a glucose and a fructose as separate molecules. When we remove a water and form a covalent bond, that's called dehydration synthesis, removing a water to form a bond. And here you see the molecule sucrose. But if we had the enzyme sucrase to come along and bind and break this bond, we'd have a process called hydrolysis, adding a water to break a bond, and then we're back to our fructose and our glucose. pH is a measure of the hydrogen ion concentration, and we use the negative log of the hydrogen ion concentration because it's easier to deal with. So instead of saying 0.000001, we say, instead of saying 10 to the minus 7th moles of hydrogen, we say the solution has a pH of 7. It makes it much easier to deal with these numbers. And we need to know what the pH is because pH determines the amount of protonation of a protein. And the protonation of the protein determines how the protein folds, and how the protein folds determines how the protein functions. I'm getting ahead of myself a little bit, but this is why we need to know about pH in biological systems. So here we have a strong acid, hydrochloric acid. We put that in water and it strongly dissociates into a hydrogen ion and a chloride, and the pH of this solution will go down precipitously. Here we have a potassium and a hydroxyl group. We add that to water, and these hydroxyl groups will sort of snatch up all the hydrogen ions, and the pH will go up precipitously. So we have a strong acid and a strong base. We add these together and we get a salt, a potassium and a chloride, and when a salt is added to a solution, it's not going to change the pH. Here's our pH scale. So notice as the pH goes up, the hydrogen ion concentration goes down, or the hydroxyl concentration goes up. Now, pH of 7 is considered to be neutral in many chemical systems. However, it's important to notice that physiologic pH is 7.4. It's a little more alkaline than neutral, and it's very important that the pH be around 7.4. Human beings cannot live outside a pH range of 6.8 to 7.8, and if your pH were 6.8, you might be alive, but you're hovering on the edge of death. So a very important part of biological systems is making sure pH stays around 7.4. we'll talk about that lots and lots as we're going through this course. So protonation is the association of hydrogen ions with other molecules. So these are some side chains I showed you. We're going to encounter these side chains in amino acids. So that's a carboxyl group. Here's the protonated form, or it could dissociate and have a negative charge. These side chains tend to be ionized at physiologic pH. But if the pH became more acidic, then we would protonate this, this carboxyl group. Here's our amino group. We can protonate it with a hydrogen, then it becomes NH3. Again, at physiologic pH, amino groups tend to be protonated. However, if the pH became quite alkaline, then the protonation would be lost. So now I've alluded to organic molecules in many, many situations, but we're going to talk about them specifically now. So we start with amino acids. Amino acids have a repeating structure. They have a central carbon, they have a carboxyl side group, they have an amino side group, they have a hydrogen, and then they have some remainder group, some group that makes them unique. So for example, we have the nonpolar leucine. We have the central carbon, the hydrogen, the carboxyl group, the amino group, but the side chain is carbons and hydrogens. So these are very nonpolar side chains. So we would expect to see a lot of leucine in a lipid-soluble protein. Now, serine is more polar. So again, we share this part of the structure, the hydrogen, the carboxyl group, the amino group, but we have a hydroxyl side chain. And since there's a big difference in electronegativity between the hydrogen and the oxygen, we've introduced a polar side chain. We'd expect to see lots of serines in cytoplasmic proteins, proteins that are in an aqueous solution. Here we have lysine. Lysine is an interesting amino acid, so we have this big long chain, but then we have an amino group, and notice that it is protonated. And so this adds interesting structure. So we use dehydration synthesis. Here's an amino acid, here's an amino acid. We remove a water and form a peptide bond between the carboxyl group and the amino group. And so as we add peptide bonds, we form the primary structure of a peptide, and the remainder groups hang off the side and give the protein its unique characteristics. We can have lots of things influencing how a long chain will interact. So here we have hydrogen bonding between an oxygen and a hydrogen. We have an ionic bond between a positive charge and a negative charge. I don't really want to get into van der Waals forces. And then here we have a covalent bond. This is a disulfide bond. So cysteines are an amino acid that have a sulfide in them, and then cysteine to cysteines can form bonds, and that influences the higher structure of a protein. So proteins can form hydrogen bonds, and then they form coils that are called alpha helices. They can also form zigzags we call beta plates. Here's how we form a disulfide bond. Then that puts a bend in the protein structure. Then we have the coils, and we have the zigzags, and we have the disulfide bonds, and we have the hydrogen bonding, and they form up into a coil. So we have secondary structure when we have the coiling. We have tertiary structure when the coils themselves coil. And if we have more than one protein coming together, I thought I had another picture, but if we have more than one peptide coming together, so for example there are 4 peptide chains in a hemoglobin molecule, then we have a quaternary structure. What kind of things are proteins doing in the cell? They're forming channels so that things can cross the membrane. They're forming transporters so things can be translocated across the membrane. They're receptors that recognize ligands on the outside of the cell. They're enzymes, so for example, adenylyl cyclase that's going to convert ATP into cyclic AMP, a second messenger. They are identity markers that tell the immune system that this cell is part of the host. All kinds of cytoskeletal elements. So we have proteins called integrins that can link the protein anchors on the outside of the cell to the inside of the cell, and also have a role in structure, but probably also a role in signaling as well. Proteins can be enzymes. So enzymes help make a reaction happen faster. They don't make a reaction that wouldn't happen on its own. In other words, enzymes don't cause thermodynamically unfavorable reactions to happen. Those reactions would happen anyway, but they lower the activation energy molecules need to form the product. So let's say we have a bunch of molecules in a solution. They're in there bouncing around, and they have to come at each other and collide in a certain speed at a certain orientation for the reaction to happen. When an enzyme is in place, the chances of the reaction taking place are much, much, much greater. So enzymes lower the activation energy. I also think about, here's my goofy example. When you meet me, you will realize I'm not a really, really athletic person. I exercise and I try to be active, but I'm never going to the Olympics. So imagine a hurdler. They have to run to the hurdle. They have to run and get up a certain momentum so they can jump over the hurdle. They have to, so jumping over the hurdle is like reaching the activation energy for a reaction. What the enzyme does is sort of lower the hurdle. So a person wouldn't have to run as fast and wouldn't have to jump as high to get over the hurdle. The enzyme is lowering the activation energy. Enzymes also provide the right orientation so that the right reactants are there. They can undergo this reaction. So this is a sucrase, and it's taking a sucrose and dividing it into a glucose and a fructose. Notice the glucose and fructose are changed, but the enzyme is still in place and could catalyze another reaction. All right, let's talk about carbohydrates. So we have a chain of carbons, or we can show it in its circular form. This is a 6-carbon sugar we call glucose, and glucose is very important as an energy source for biological systems. If we just have one glucose, or here's a galactose, if we just have one sugar, that's a monosaccharide. But using dehydration synthesis, we can take two monosaccharides, turn it into a disaccharide with the removal of water. If we add the water, we can break up the bond, and that's of course called hydrolysis. And then we can form long chains of glucose. And when we take long chains of glucose, we can make different kinds of molecules. We can make a molecule called cellulose. Plants make that as a structural substance. Plants can make starch. We use that as a food source. And then animals make a sugar storage molecule called glycogen. They're all made out of glucose, but the carbons that bind and the enzymes that are required are needed. But they're all complex molecules made out of glucose molecules. All right, here we have some lipids. So this is a fatty acid. So notice long chains of carbon after carbon after carbon after carbon. This is very, very nonpolar. We have a little carboxyl group at the end, but very long chains. These are nonpolar molecules. Notice we have one that's saturated, all single bonds. So when carbon is bound to 4 things, the carbon has been saturated. Notice in this polyunsaturated fatty acid, we have some double bonds. So carbons are sharing a pair of electrons, And so in this case, the carbon's bound to three things, so the carbon is not saturated. The significance of that is saturated fatty acids tend to be long and straight, and so when you take those fatty acids and make a triglyceride, which is a glycerol backbone and three covalently bound fatty acids, the long chains fit very nicely together and they will form a solid at room temperature. So that tends to be animal fats. If you have a lot of carbon double bonds, that puts lots of bends in the saturated polysaturated fatty acids, and then when you make a lipid out of it, make a triglyceride, it has lots of bends. They don't pack in together. They tend to be a liquid at room temperature, and those tend to be your plant oils. And so it's much harder to make cholesterol out of a polyunsaturated fatty acid than a saturated fatty acid. One reason why the plant oils are more heart healthy. And is there something else I wanted to say about that? It has escaped me. I guess we'll move on. All right, here's a phospholipid. So we have fatty acid tails, and then we have a polar head. So here we have looks like a glycerol group, and a phosphate, and then some other thing. A sugar, could be choline, could be a inositol. Anyway, thermodynamically, these will spontaneously form a bilayer, sometimes a vesicle, but here you see a bilayer, and the tails are together and the polar heads are sticking out. That maximizes hydrogen bonding in the aqueous compartments. So this is the lowest energy state for this arrangement, and so phospholipids will just spontaneously form bilayers. That's the lowest energy state, and it maximizes the hydrogen bonding of the system. So again, here we see some amphipathic molecules, and I believe I mentioned that word a little bit ago, but when you have a polar region and a nonpolar region in the same molecule, that molecule is called amphipathic. And the lipid-soluble structures will form together. Here you see the lipids in the center. Here's the polar heads. They're all hydrogen bonding with water. So this is the lower energy state, and they maximize hydrogen bonding when the lipid substances collect together. And you can see this with just regular lipids. If you're making spaghetti and you add a little oil so your noodles don't stick together, the oil all collects on the top. Why is that? Because that maximizes hydrogen bonding in your water. Another kind of lipid is a steroid. Steroids are derivatives of cholesterol. Cholesterols are highly, 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 highly hydrophobic, except they have this one little hydroxyl chain, and I'll show you the significance of that. So technically you could say cholesterol is amphipathic, though it really is a lipid-soluble molecule. So cholesterol can have some structural functions. We also can change the side chains to make other molecules that are called steroids, and steroids are very important as signaling molecules. All right, we're in our last section, our high energy compounds, so nucleotides. So here we see a base, a cytosine, here's our sugar, deoxyribose, and there's the phosphate. So this is deoxyribose nucleic acid, and you put a bunch of those together and you make DNA. Here the sugar is ribose, you put those together and you make RNA. And so here you see the base pairs forming a twisted ladder, or the double helix. C, hydrogen bonding with the G, and the adenosine hydrogen bonding with the thymine. We also talk a lot about a high energy compound called ATP. So adenosine trisphosphate. So adenosine and ribose and three phosphate groups. And And when we have the 3 phosphate groups, this last phosphate, this is called a gamma bond. And when you break that bond, you release a large amount of energy that is just in a perfect form to run biological processes. It's just the right amount of energy in the right form to form a peptide bond, to pump an ion, to make a muscle contract. It is a wonderful form of energy, and maybe in one class you heard ATP is the currency of the cell. Why do they say that? It's a form of energy your cells can understand. Your cells don't understand donuts. They don't understand donuts. You have to break it down into glucose, and the glucose has to be made into ATP, and ATP is the substance that your cell can deal with. It's a substance that your cell can use. We'd also include FADH and NADH in this category. We'll talk about that on another day when we talk about metabolism. So here we are at the end of the presentation. Think about questions that you might have to bring to class. Go back to the beginning. Look at those learning objectives. Can you explain those learning objectives to your cat or to your friend in the class, or can you write them down on a blank piece of paper? If you can, then you know what you need to know for the assessments coming up in class. Thank you very much for your attention. I'll see you next time. Bye-bye.